DNA replication has a double helix structure, with two complementary strings intertwined together. When chromosomes divide during cell replication, the strings are unwinded by an enzyme called helicase. DNA polymerase then adds complementary nucleotides to them, following the rule A to T and C to G. Polymerases can only synthesize from 5' end to 3' end direction using an RNA primer created by primates that initiates replication. On the leading strand that is oriented from 5' end to 3' end, the polymerase is able to continuously replicate from the beginning to the end. The lagging strand, however, is oppositely oriented from 3' end to 5' end, so polymerases can only conduct discontinuous replication by repeatedly synthesizing primers and creating small fragments of DNA. These are called Okazaki fragments, and later combining them together. The primers will be then removed and polymerases can refill their gaps. However, the last primer for the lagging strands sit at the 3' end of its copy, which is 5' end of the original strain. The DNA polymerase cannot reconstruct this part by moving in the wrong direction of function. Therefore, a small part of the original DNA is not copied. In order to protect important information from being left out, an extra length of meaningless G-enriched code is attached to the end of chromosomes, so they will be lost during each replication instead of the essential materials. This extra part of code is called telomeres. As chromosomes replicate, the length of telomeres decreases until used up, and essential genetic materials will all be exposed to danger of loss in further replications. Cells will no longer be able to replicate, therefore approaches senescence and cell death occur. This limit of replication is called the Hayflick limit, which cells will reach within 40 to 60 replications. In order to support more cell divisions, there are enzymes called telomerases in stem cells and gametes, which can increase the length of telomeres when used up. Telomerase is an enzyme that can bind to telomeres at the 3' end of the lagging strand. It carries a piece of RNA string and uses it as the template for extending telomeres. Nucleotides can be added to the lagging strand accordingly as the RNA piece binds to the end of telomeres. This process is repeated in order to restore the length of the telomeres, and complete DNA information can be protected during further replications. Telomerase activity is strictly controlled in somatic cells. However, mutations would occasionally occur in cells and produce a high level of telomerase. This characterizes the transformation of normal cells to cancer cells, because such high telomerase activity allows them to repeatedly elongate their telomeres, so they can unlimitedly replicate, therefore expanding and forming tumors. In contrast, another group of diseases are caused by the downregulation of telomerases, known as the telomerase biology disease. These diseases are characterized by extremely short telomeres, which impede tissue cell mitosis and lead to tissue failures. Symptoms of it include abnormal skin pigmentation, dystrophic nails, and bone marrow failure.
Recent studies show that telomere length can also affect the pace of aging. While there are many external factors like economic status and smoking, the size of telomere inside of body retains its strong association with human lifespan. Telomere length decreases at a rate of 25 to 28 base per year with age. When its size reaches below a critical limit, the cells undergo apoptosis, a natural biological process of programmed cell death. In this stage, the cells would get the signal to conduct self-destruction for the smooth functioning of the whole body. Consequently, as the shortening of telomere goes on, our cells will become less efficient in performance and slowly die out, or in some cases have genomic instability through mediating interchromosomal fusion and possibly cause cancer. For example, as we get older, the pigment cell in our hair follicles gradually die. When there are fewer pigment cells in a hair follicle, the strand of hair will no longer contain as much melanin and will become a more transparent color, like gray, silver, or white. Furthermore, the rising risks of cancer in elders also supports the effect on aging by telomere length.